this is the slide we're starting on. We're talking about how things get through that membrane, and if you're really big, like a protein, you got to be transported across accidentally. You don't go by yourself. You got to be forced across. And things have to go in the nucleus, and things must come out of the nucleus for two reasons. One thing, to maintain the nature of the chromosomes. The sun outside there can mess up chromosomes and cause skin cancer. You know that, right? Now, there are some folks in here who would have skin cancer had not been for the maintenance of, so you're fine. But the more you work outside, the more you put stress on it. My whole family is a tree service. They work outside all the time. And I'm just amazed that none of them have any skin cancer yet, because they work out there all their lives. And when I lost my leg, I stopped working. I take my shirt off to work. Because if your shirt got wet when you get dinner, it was kind of unpleasant. So got a nice tan. But by the same token, when you got that nice tan, you're also exposing yourself to UV radiation. But luckily, your chromosomes have the facility to maintain themselves and fix damage. And even this, if the area is so badly damaged it can't be fixed, it's isolated like in quarantine. And then new chromosomes. That's why, remember I asked you one time, does every single part of a gene count? No, because some parts get isolated, figure it's too bad to fix. We'll make more of the stuff. And this part we'll just tend to ignore. So every single place on a gene does not mean something. A lot of places do. And gene expression, you know, blue eyes, brown eyes, skin color, even your ears. Some ears come down and go back up. Some go straight in. That's all gene expression. The ears that go straight in are recessive, and the ones that kind of hang down, that's dominant. Some folks can roll their tongue like that. That's dominant. I play harmonica. And I had a person one time ask me if I would show them how to play harmonica. Fine, but I said, can you roll your tongue? And they couldn't do it. I said, well, that's going to be a problem, because after you learn to play that, you can use what's called tongue blocking, where you block it with a flat tongue. But those holes are real close together, and you want to play a pure tone, you must have air go down one hole and not the three together. If you roll your tongue, you can direct air down one hole, or you can draw air through one hole, and that's how you play the harmonica. When I first started, I had a little book explaining, you know, they have numbers on top of them, 1 through 10. And if you're going to play like America is Beautiful, it would have arrows. Number 1, arrow this way, 2, arrow that way. And you just, it'd be very mechanical, but you could play it. And as you got better, you put the book down and just played the music. And at this point, I have a harp in my car right now. I'm always playing. The radio ain't got no work crap on. I play harmonica. And I make tunes up. And I'll play things that I remember, like I like to play Amazing Grace. I like to play Shenandoah. That's always a pretty song. I like to play Dixie real slow. I was one day at Hardway playing Dixie, and a black person came in. I thought, oh my God, I'm in trouble. And the girl says, that is so pretty. What is that? And I went, it was just something put together. I never told the Dixie. Matter of fact, Dixie is written by a black person. Did you know that? A black person wrote Dixie, tongue in cheek. Oh, I wish I was Atlanta Cotton. No, she didn't either. But she wrote the song, and a lot of folks don't realize that that's not like that one or something. That's a black song, but it's so pretty. I don't like it. I can play it fast, but to me, the beauty leaves it. If you play it real slow, it's a real pretty melody. And I don't like to say the words. I just like to play the tune. And now some folks recognize it, and some folks, when you play it slow, they don't know what it is, because it's so pretty slow. All right. Inside the nucleus, there's no vacuoles, there's nothing inside there that has a membrane wrapped around it. Even the nucleolus has no membrane. It's just a bunch of, it's like a mole. It's just a dark area. And I mentioned, of all those, what do you think it means to be subnuclear? Does it mean to be under the nucleus or smaller than a nucleus? Smaller. It means smaller. I know a submarine goes under the ocean, but in this sense, sub means smaller than. Like the subatomic particles, electrons, protons, they're just smaller than the atom. Well, subnuclear means smaller than a nucleus, and the nucleolus is inside there, and it is smaller than a nucleus. It's a subnuclear particle. Some nuclei have more than one nucleolus. Some have just one. Some have little blotches of them. But the nucleolus is busy making ribosomes that go outside and the cytoplasm, and then they get busy making proteins. And you may need protein. I mean, your hair is protein. The iris, that's protein. I mean, you can't get away. You know that those receptors in the cell membrane, they're protein. Ribosomes are so busy all the time making structures which are made of proteins. And like I said, exported means to be sent out. And they're so small, they just move right through. Now the messenger RNA has to be pumped out. But he gets pumped out. But the ribosomes, they have one claim to fame, and that is they make the proteins. Nothing else makes your proteins but the ribosomes. So, well, I thought the rough ER did. It does because it has ribosomes. That's why it's called rough. I mean, the ribosomes make the proteins and put those in the ER, and they move away like a train on a track. But that track ain't making the proteins. The ribosomes are making them still. All right. This shows you, and both of those are real pictures taken through a scope. Wrong way. Well, this is the cell. This is the nucleus. Pretty big nucleus too. And this is the chromatin. It's like spaghetti. It's not condensed chromosomes. And that is the nucleolus. Now, if you blow it up real big, here's your nucleus. All the stuff out here is cytoplasm. And this is not in the membrane. It'd be more, if it's in the membrane, it'd be a sharper edge. But this is the nucleolus here. Now, in my picture, do you see any of those pores? I saw some. I just looked at it. Right there's, I see a good one right. Let me this thing here. Right here, I see one. I see one there. I see one there. I see one there. They got little arrows. Looks like pointing to them. And that's how stuff comes in and goes out. There's no pores in your cell membrane. They have those proteins which open and close doors. But the nuclear membrane, they have just, they take the door off the wall and you come in as good as you want to. They have pore. So there's no there's no proteins in that membrane that's going to control. That's the open door policy if there was one. If you're small enough, you can go in. If you're big enough, like a big old mitochondrion, it's not going inside there. It's just too big. You can't get to the door. So there's the... Now, a lot of folks will tell me that is the cell, and that's the nucleus, and they don't know what they're talking about. I have blown up so big, and you're inside the cell, and you're looking at the nucleus. Now, the one over here, I'll show you the cell also. But this is the cell. And there's your nucleus. And I just show you pieces. What do you reckon that stuff right there is? That's the ER. I don't see any piece. I don't see, that might be right there, a piece of a mitochondria turned this way at you. But this is all ER. That's the railroad tracks that carry a thing around that cell. All right. Little history. The first structure in a cell that ever discovered was that big old nucleus. It is an organelle. It's the biggest one you have. And far, 17, that's a long time ago. Antonie van Leeuwenhoek, when he saw it, he called it the lumen. Well, a lumen is a hollow structure inside a straw that your coat goes up. That's a lumen. Inside your blood vessels, the hollow that the blood goes through, that's the lumen. The nucleus is not hollow. So to call it a lumen really is not correct. And that's why we changed it. Now, red blood cells in salmon, they have a nucleus. But your red blood cells do not have a nucleus. When they're born, they do. But as they grow up, the nucleus goes away. And the reason is, a nucleus, by nature of activity, consumes a lot of oxygen. It's got a tremendous appetite for oxygen. Well, what's a red blood cell carrying? Well, if he had a nucleus, when he got to where he's going, what would be the problem?
He would take some of it.
The nucleus would have already eaten all the... And when he shows up, he's empty. So the way the body solves it is no nucleus. But then the problem is, how long can a red blood cell live?
Not long.
Not long at all. And can a red blood cell make more red blood cells? No. Your bones make all your blood. Now, when that blood cell is first born, it has a nucleus. But as it grows up, as it matures, because its job is going to be to carry oxygen, and I have a thing inside me that eats oxygen, it behooves me to get rid of it. And that's what they do. We're not sure why salmon still have their nuclei in there, unless they don't need much oxygen. I suspect that's a problem. Salmon live in hot water or cold water. But cold water make you have high metabolic rate or low metabolic rate. No. So if you have a low rate, do you need much oxygen? No. Now if he lives in the tropics with a high rate of metabolism, what would be the problem then? The nucleus would be, he'd probably die. But where he lives, because it's so cold, he can get by with it because he doesn't have a high rate of metabolism to start with. But you have a high rate of metabolism, and though you have 3 to 5 million red blood cells in a drop of blood, it sounds like a lot, doesn't it? But they're so small, it's not much at all. Matter of fact, when I say that, my mind thinks about a cubic millimeter. And if you want to know how big one of those is, take a dime. Make you a piece of wood square. All the sides are a dime thick. How big would that square piece of wood be? If every side was a dime thick, how big would that be? Would it be big or small? That's a cubic millimeter. Now, if you have a drop of blood come out of your finger, is that drop of blood more or less than that cubic millimeter, you reckon? No. It's more.
It's more.
More. And I tell you right now, we know for a fact that in a cubic millimeter, and that's the square size of a dime, there are between 3 and 5 million red blood cells. So how small do you reckon those things are? Well, they're tiny. White blood cells you have like maybe 3,000. They're big. 3,000 though in that drop is still a lot. They're still small. Platelets you have like 340,000 platelets. But they're not whole cells either. They're pieces of cells. And if you cut yourself, they start the clotting process so you don't bleed to death. But the problem is though, inside your body when the blood's being pumped around, you don't want clots there, do you? That's how you get a stroke. So heparin shuts down the platelets, so inside your body, they can't mess you up. But when you cut yourself, shaving or whatever, you then want those suckers to work to make the bleeding stop. And then the heparin backs off and the bleeding stops. When they had my leg cut off, they were concerned that as it heals, it's going to make a lot of blood clots. And those clots, if just one of them got in my lungs, it would kill me. And so while I was in there, they gave me a blood thinner. They gave me lots of heparin. And they took my razor away from me. Now, were they afraid I was going to cut myself and bleed to death? No. I was going to mess with the bedclothes. I was in no danger of dying, but if you cut yourself and you got tons of heparin, it's going to bleed and bleed and bleed and bleed and bleed. You got this to make your pillowcase, it's going to be on your robe. It'll make more work for the nurses. So they took my razor away from me, and if I don't, I shave. If I don't shave, I don't feel clean. I can take a bath and not shave, I still feel dirty. And in the hospital, I didn't feel good because I felt dirty. They would wash me, but I feel my old rough beard. I said, my hair got long. And they put me in a wheelchair and took me down to the barber shop. Just cutting my hair made me feel so good. And my son, and my older son, I think actually had it, they bought me my first ever electric razor. I didn't think I liked that thing. You know, I love it. I bought me another one. I bought me one by Braun, and that bad boy was like $300. So I went big time. I went from the $39 special, the one that, when that thing shakes, when I get done, I feel as close as a blade. They said it would. Got paid for it though, man. And I love it. And my beard is kind of heavy. I didn't think electric razor would do much good. And I still, and that when they bought me, has now broken. I still have it for a keepsake. I don't want to throw it away because that's a good memory that they bought me that razor. My two sons did. All right. These are the pictures that Antoine drew in 1712. Now this is 2011. How long ago was that? Roughly how far back? Do your subtraction. About 300 years ago. That's what he drew. As a matter of fact, this dude was the one who, he didn't invent a microscope, Robert Hooke did. But he did make he was a lens grinder. He made the very best lenses in existence back then. So, because he looked at so many things through a microscope, he is called the father of microbiology. Anton, and it's not Antonius, we call him Anton. He is, back in his day, he was the man. Now his scope can't hold anything to ours now. But back then, this thing was top of the line. He was the first microbiologist to carry that label. Now, Robert Brown was the one, more than 100 years later, who first described in great detail the nucleus. Now he had a better scope too. Now he could see better with it, and he was the botanist. He liked to look at flowers, orchids, and he was the one who used the word nucleus for the first time. So the word nucleus is Brown's. Before him, Lumen was the word used, which was van Leeuwenhoek. But when he saw this, he said, "I'm going to call that the nucleus." And the word nucleus, it means kernel. And a piece of corn, there's a center inside it, and it's full of water. When you heat it in a microwave, that thing makes steam and explodes. That makes popcorn. Now, why do some kernels never pop? Because the water leaks out of them. And I know Oral Redenbach, when I buy it, because he has, you pay more for it, but his kernels, now some will still won't pop, but more pop than not. I don't want to have half of them pop me till they're half away, so I pay a little more for it and get, but even his, I don't know which is the worst brand to buy, but I don't mind paying money for something that's good. Well, to a degree, I ain't paying $500 for it. All right. So Leeuwenhoek was the first one to see it, but he called it Lumen. So he was the first one to draw pictures of it. Robert Brown came by 100 years later, 115 years later, like that, and said, we're going to call it nucleus, and it stuck. He wrote the papers, he made the books. He called it nucleus, so we picked up on that. If Leeuwenhoek had said nucleus, it'd probably been named back then. All right? Now, Schlei, he figured out that this nucleus plays some role in cell division. And he said this nucleus is a cytoblast. In science, the word blast means to build. It doesn't mean like to blast and destroy. It means to build. Class means to break down. Like, for example, your bones. If you break a bone, osteoclasts come out and dissolve that broken bone. But they save the pieces. Osteoblasts come later and take those pieces and reassemble them and heal your bone. And then when the osteoblasts are finished and your bone is healed, they turn into what we call osteocytes. And they live there just maintaining it. But the osteoclasts are the ones that actually, you've got to remove the broken bone before you can fix it. So the osteoclasts come out and they dissolve the broken bone. Don't throw it away though, they can use that stuff again. But they can't build. So when they get done dissolving, in comes the osteoblasts and take all these things and recycle, and they start making new bone. It takes about a month, as you know, how long you wear a cast. And then once they get done making new bone, they paint themselves in. When they're done, they're trapped. And you recall the little kindergarten show about the steam shovel in the building, and he couldn't get out. They built the building around him, so they used him as a furnace to heat the building later on. Y'all don't hear kindergarten stories like that? No, I can't believe it. By a little train that thought he could. You heard that one, right? Yeah. But the steam shovel was another one where they built the building with no thought of, let's leave a place to get him out of here. When the building's done, he is, and you go, oh my goodness. So they made the best thing of it, made him. So these osteoblasts, they build the building around themselves. And when they get done, they go, oh crap, I can't get out. Well, the body says, fine, you're going to stay here and serve you the rest of your life as a maintainer. And we're going to call you now an osteocyte because your building is over. So cytoblast is any builder of cells. Okay? And he thought, he saw new cells coming from the nucleus, he thought wrong. He thought wrong. But the nucleus does control when the cell divides. He thought right on that. Way back then, they had some strange ideas. They were just their best guess. And sometimes the best guess right now is laughable. You go, how could he have thought that? Well, he had nothing else going. We look back on our knowledge, you go, that was stupid. They thought that dead meat could turn into flies. Because they saw flies coming from dead meat. What they did not know was that flies land on that meat and lay their eggs. And then those eggs hatch out the larva, and then the pupa, like you saw upstairs. And then the pupa, they turn. Well, they didn't realize that. Why not? Why can't dead meat turn to flies? And the meat slowly goes away. And they thought, well, it's turning to flies. Well, Louis Pasteur, the one who does he shot that sucker down. But Spallanzani was the one who got all the criticism. When Spallanzani first said that ain't right, they thought he was an idiot. Who gave you a degree in science to be a teacher? Spallanzani was exactly right, and Pasteur proved it. All right. Now there's Meyen. Now, remember now, Schlei thought the nucleus could make new cells. Meyen said, no, it can't. Because Rudolf Virchow had done proven that cells come from cells, not from a nucleus. A don't know how Schleiden forgot that. But Schleiden, so old Franz come along and now do they know each other? Now look at his birth, look at his lifespan. Schleiden, he died in 1881. This bad boy, they were contemporaries. They knew each other. Of course, this guy died first.
Yeah.
This guy died 40 years later. They're both born though, look at that. In the same year.
How does that?
I get one of them a smoke.
Yeah.
I don't know why. But he felt that the idea of a nucleus giving rise to a cell was just stupid. Because Virchow had been proven that cells come from cells. And as a matter of fact, Virchow's thought that cells from other cells actually now is part of the cell theory. The cell theory says two things. All living things are made of cells, and all cells come from pre-existing living cells, not a nucleus. Not a nucleus. So, and Meyen was right. Schleiden was smart, but he was wrong with that number. He was wrong with that number. We look back now, we can see all this stuff, how it was so wrong. And back then, though, in that day and age, you know who was right or wrong. I mean, Schleiden had a high reputation. Schleiden fathered half the cell theory. Meyen didn't father anything. So, just by pure fame, you would have thought, well, Schleiden must be one right. This guy here is an upstart. But this guy here was right, and the big scientist was wrong. Okay?
So, if Schleiden fathered half the cell theory, and then Virchow came back and just created a new cell theory, or he...
No, okay. Let me back up. Okay.
Reiterated?
Matthias Schleiden was a botanist.
Right.
And he studied plant cells. And he never saw a plant cell. He never saw a plant that wasn't made of cells. So his half said, all plants are made of cells.
Okay.
Now, in the same day and time, a guy named Theodore Swan, I remember because Swan's a bird, right? Well, he was a zoologist. And he studied animals, and he said, you know what? I ain't never met an animal that wasn't made of cells too. So when you put those together, what do you get? All living things. Because back then, that's all there was. We knew about viruses. We knew about plants. Well, if Schleiden is saying all plants are made of cells, here comes Swan saying all animals. Can't you then say all living things are made of cells?
Yes.
Yeah.
Well, that's how. See, half the cell theory came from Schleiden saying all plants are made of cells. The other half came from Swan saying, well, guess what, Schleiden? All animals made of cells too. Therefore, I guess all living things made of cells. And then Virchow came along and said, and one more thing, guys. One more thing. All cells come from other cells. So all three men are given credit for the cell theory. Schleiden said all plants are made of cells. Swan, which is S-C-H-W-A-N, he said all animal made of cells. And Virchow said all cells come from other cells. They can't just spontaneously erupt like flies or dead meat. All right? So those three guys are the fathers of the cell theory. Now the cell theory has two components. All living things are made of cells. And all cells come from other cells. Well, how do you get three guys with two components? The first component can be thought of as two ways, right? All animal made of cells, all plant made of cells. Put them together, what do you have? There you go. So, Schleiden and Swan, they give him credit for the first component of the cell theory. And the second component, that all living cells come from other cells, that's Virchow's baby. But all three guys, right now, they're the ones getting credit for stating the cell theory. That answer your question?
Yes.
Okay. Now, the last slides are going to talk about the chromatin, because there's no way I can end this PowerPoint without describing you how chromatin shrinks up and becomes chromosome. So here we go. Chromatin is made of two things: DNA, which is your genes, and proteins that they're wrapped around. All right? If you had a long ball of twine, some folks would take a stick and wrap it around the stick, so it don't just fall apart, right? Well, the twine you wrap around would be the DNA. The stick would be the protein. All right? Now, when you wrap this stuff around, we call that a histone. The DNA wrapped around the protein, that's a histone. I made a picture, I believe, of that. Now, in every cell you have, there's about 6 feet of DNA wrapped all around to make the histone. And that 6 feet is what produces your 46 chromosomes. Now, half your chromosomes came from your mom. Half came from who? Dad. Now, if half came from mama, how many sheep did she give you?
23.
How many dad give you?
23.
Now, the one from mama that controls hair color, and the one from dad for hair color, they come together, and the dominant one will dictate, right?
Mm-hmm.
Now, the two, they're called homologous. They're homolog because they both control the same trait, right?
Yeah.
Now, if the next one comes out and controls maybe if you can or cannot produce insulin, you got one from mama, one from daddy. They come together. Those are homologs. Now, will this one pair up with the one above it? No, because they're not homolog. So a homolog is the one from mama and the one from daddy that control the same trait. And that's what we say you have 23 pairs. We almost never say you got 46 chromosomes. We say you have 23 pairs, which really means 46, right? But you got to remember, they come one from mom, one from dad, and everything about you is controlled by your chromosomes. And you might even look like your grandmother and not your mama at all. But guess where your mama's chromosomes came from? Her mama. And that's why it might pass right through her and come into you. And you might have pictures of your grandmother when she was 16 and got doggone. I look like grandma does. Because the DNA passed down the line. Yes.
What if you look like your uncle?
Same story. Your uncle is your brother, is your father's brother, right? What does your father and your brother have in common? Their parents. It's very likely that if you like your uncle, then in my household, my side are the Edwardses, and they're Woods people. They all got the belly, like I'm getting, and they're hitting hairlines. On my wife's side, they got no hair, and they're all short stature people, but they're all muscular. Now, if you look at my two sons, you're going to see both sides of the family in there. My youngest son was a great wrestler because he was small, compact, and powerful. My older son was a great runner. He was long. And I can look at both my sons and say, I see my mama right there. I resemble my mother and my complexion. My father has a vicious temper. My sister does, and I'm easygoing. I mean, nothing to. They go, don't you care about this? Yeah, I care about it, but not enough to throw things against a wall. No, don't throw things. If you're in the way, tough. See, yeah, they assume if I don't go into a tantrum and stomp my feet and yell and scream, I don't care. That's not my character. I tell you what, though, when I get mad, you better get out of the way. So when my fuse is finally lit, you've gone too far. And I was a boxer in the Army. And they said, Jeff, you got to get aggressive. Well, I ain't mad. But that guy hit me one time. I get mad then, buddy. And I was undefeated till my last match. And that guy hit me in the chest. And they always told me, don't, when the bell rings, you drop your arms, right? And you go to your corner. They said, don't do that. Well, I didn't listen. And I was 82nd, I was boxing some guy from the Rangers. And I had him going pretty good too. I couldn't knock him out, I hit him once. And when the bell ended the second round, I dropped he had a punch come hit me right there. I could not breathe anymore. I couldn't breathe anymore. And all I did was, I was kind of shadow boxed. Don't knock me out. I lost the match. He went to Germany and fought some dude's punching forces and got knocked out in 3 seconds. I was so glad I lost that match. I thought, God, that could have been me laying there all splayed out. So I reckon the good Lord would watch over me and said, no, Bobby, you don't want to go to Germany.
What's the telomere?
Huh?
You said it said one chromosome attached to the next at the telomere.
They're in a line.
He's like this and
They're in a line.
he's looking up there.
Well, how can I show you? You know a freight train out there? How they're hooked together? With that couple with the hook at? That's the telomere. And the boxcar is a chromosome, a telomere, or a chromosome, and they're all hooked together like that. So the telomere, tele means terminal. It means the end. So the end of one chromosome hooks on the next chromosome like two freight cars hooked together at the coupler right there. That's the telomere. And this nucleus, now you got to know now, 6 feet is crammed in that little, come on, somebody packing package. That's packing right there, buddy. All right. I ordered 91 pieces of track for my hobby, and it came in a little box. I went, how in the world? Opened the box, and I took the track out. I can't get back in that box. No. I said, who packed this thing? Because he's the world's best packer. I said, you do Christmas wrapping too? I'm going to hire you. I took that track out, 91 pieces in a row, and I got maybe half of them back in there. I'm out of room. I go, how in the world? When I thought about the Nucleus, how well he is. Someone's packing him really good. Okay, go. All right. So the Nucleus packs as good as that guy packed that track. I can't get in. I can't get in it, so I couldn't sell it again. I can't get back in the box. All right. See the ends of them? See the white ends? Those are the telomeres. That's the end of the chromosome. Now, why is that not called chromatin? Because I can see the individuals. When you see them individually, they're chromosomes. And when they lengthen and lengthen like a big bowl of spaghetti, I can't find one from the next one, that's called chromatin. Chromatin shrinks up, and starts like that. And the ends of them, where they would attach later on, those are the telomeres.
So that would be the chromosome?
Yeah, I see a number on there too. I don't know if I got 46 there. I haven't counted them. I didn't even want to count them. So
at what point do they look like this? I mean,
when they're in the cell, like that, yeah, before cell division occurs. They must shrink up like this for one reason, for the cell divide.
The chromatins and stuff,
that's not chromatin.
Huh?
That's not chromatin.
No, I'm saying the chromatins, is that where it looks like an X?
No. That's where it looks like a bowl full of spaghetti.
Where it has all the lines?
No, you're confusing cell division. You know interphase, when nothing's happening, and he's just living? That's when you have chromatin. The cell grows bigger, now it says, well, I got to divide and get too big. Well, before it divides, the chromatin shrinks, because I got to pull it out of the way. What's easier to pull, a long one string spaghetti or something that short?
spaghetti.
Well, that's why I get short. Because when that cell will divide down the middle, you can't have any strands across it, but they're going to be cut in half. So by being shrunken up, they get out in the middle, and then the plate comes across that cleavage. Then now, once the cells are divided, and we don't need it thick anymore, they get thinner and thinner and straight and straight and straight. I didn't one day walk in with a bowl of cooked spaghetti and ask you, Tim, how many spaghettis in a bowl? Could you do it?
Nope.
By just looking? You can take them out. Now what you could do maybe is weigh one piece of spaghetti by itself, right? Well, you got to weigh the bowl too, right? Subtract that weight, and then, so and one piece of spaghetti weighs 1 gram. And I have 100 grams worth, that'd be, you think that, unless all different shapes, different sizes. Now, there's your X. Now, let me show you something. You see in here, like the bowl of spaghetti? I can't see this, can I, in here? But they start shriveling. And they start shriveling. And before long, you have the double helix. Now, this is smaller. This is smaller. Getting a little bigger. Starting to wrap on itself like me wrap the twine. Getting a little bigger. Nice encased in a fiber. And this, all of this, is this. If I magnify this, I will get this. If I magnify this, I'll get that. If I look at the real high magnification, I'll see that this is composed of the DNA. So when you go from small to big, this is tiniest. They're starting to wrap around the proteins now, like they wrap that thread around a bobbin. Now they get a little bigger and they start to wind back on themselves. Like rubber band, you wind up a little airplane, how it winds itself, they start getting real knotty. And then you have a covering on it, and that's why it's called chromatin. Now, this will start getting thicker and thicker, and before long, it'll look like this. There's one end, and there's one end, and there's one end, and there's one end. This came from mama. That came from daddy. And where they're attached is the centromere.
So you're saying that chromatin
will condense to look like this. Does this look like that? No. But you keep wrapping this stuff up, wrapping it up. Before long, you're going to have it looking like this is a chromosome. Actually, that's chromosomes are replicated. Because when they divide, this thing, I don't want to get too far ahead of myself. I'll confuse you more than you already are. But when you see it like this is a chromosome. We call that a chromatid, and that's a chromatid. When they separate those two chromatids, they get to be called chromosomes then. And one goes to one new cell, and one goes to another new cell. Both those came from one parent. So, how many X's will you have? If you have 46 chromosomes, how many X's will you have inside there?
23.
46 of them. They're going to do that. And actually, does that represent one chromosome or two right there? That's actually two right there. And when they separate, they become individuals. And that's it. Now, that's ahead of me in my. I got to do mitosis, and you'll see more what I'm talking about. But right now, just understand that the chromatin is a non-condensed, very, very fine, fibery. You can't tell one from another one. And as they get thicker and thicker and thicker and condensed, you start to see them then. And then you stop saying chromatin and you start saying chromosome. I don't know where a change occurs. At what point do you say, well, I can now call it chromosome? I don't know where that is. I mean, it's like going from here to across the river to Alabama. At what point are you in Alabama? Actually, the river belongs to Georgia. Alabama's mad about it too. If you come in to Columbus from Alabama, the Columbus city limits is on the Bama side of the river. The river belongs to Georgia. Now you would think of it down the middle, wouldn't you? I don't know who did that by politicians, but when they divided, somehow Georgia got all the river. Now, as it backs up the sloughs, that's Alabama. Now, if you're in there fishing in a slough, you have your Alabama license. If you're in the river fishing, then your Georgia license. And a fisherman knows what license they have. A lot of folks have both licenses. But somehow, when they said who owns the river, Georgia snookered Alabama. Like I said, when you come, I would think the line should be in the middle of the river. And he that one. The Georgia line is on the Alabama bank. The river belongs to Georgia. And of course, Bama's still mad. I don't know how they're going to do it because sometime way back in old days, the Bama politician was asleep when the Georgia guy drew the line. River's mine now, Jack. They're still, and Florida, they're mad too. Florida ain't got none in the river. So you got 3 states bickering. Now, it's not just thrown in there because you're going to find that the one from mama somehow can find the one from daddy, and they get side by side. They pair up. Now, girls, boys, which pair, the first or 23rd, determines your sex? Do you know that?
You died this
The
one.
last pair. The last pair determines the first 22, hair texture, skin color, whether or not you're going to have breast cancer one day, whether or not you can roll your tongue, all those things that make you you, that's the first 22. The last pair, all right? Ladies, you have 2 of the same kind. You have X and X. That's a long chromosome. Guys have X and Y. And X and X means tall and tall. X and Y means tall and short. Now, when my wife makes an egg, she can put in one of the X chromosomes or the other one, right? So who determines the sex of the baby boy? Or
The
girl?
man.
The man does. If I make sperm, this sperm gets my X and this gets my Y. Okay? The wife has only what kind? Well, if this sperm, which is X, fertilizes that egg, which is X, then that becomes XX. That's a little girl. If this sperm, which is Y, finds that X, that's a little boy. You know, King Henry killed that woman for not giving him a son. It wasn't his fault.
That's
It wasn't her fault.
Developing countries.
But back then, they know that back then.
My friend just
They
died.
thought the lady was one who gave children. So if you can't give me a son, woman, that's your fault. No, it's not.
It still
She
happens.
can't. All she can do is provide the X chromosome. He's got to provide the Y. But back then they didn't know that.
They don't know that. They still don't know it.
Now, there are things on the X chromosome that get a boy in trouble, like hemophilia. If this part down here is not matched by the Y, and that says hemophilia, what's going to stop it? Nothing is. Guys lose their hair. That's on the X chromosome. Male pattern baldness comes from the mama, not the daddy. Because how much hair I have is on the X chromosome. And my little son got my Y chromosome. Nothing on there to control his hair or his blood type. It's all from mama. As a matter of fact, your skin, what you feel right there, how big is a sperm compared to an egg in size? Which one is huge? Egg. You know why? The egg contains a nucleus and cytoplasm, right? Mm-hmm. The sperm only has one thing, a nucleus, no cytoplasm. All that sperm does is, that's the way to contribute my half, my DNA. Now, when my DNA enters that little egg, and if my Y is there, what's the baby going to be? A boy. If my X is there, it's going to be a girl. Now, all the mitochondria, all the reticulum, all the cytoplasm, is that from me or from mama?
From mama.
All your stuff, it's from your mama. Dad didn't give you any cytoplasm whatsoever. So when that egg began to divide, you just kept what Mama gave you. So you can look at your skin and look at your mama. Because your skin texture is from your mama.
So what
All your cytoplasm is from your mama. Not one dab came from dad.
So what does the father contribute then?
Just the 23 chromosomes. That's it.
And the sex?
That's all he gives him. That's all he contributes. But now, if he don't, what's going to happen to the egg? He's going to die. I've got to give my chromosomes because to be a human being, how many must you have? 46. Well, the wife has got 23. I provide my 23. The baby then has 46. But all I gave that baby was one thing, my DNA. I didn't give any cytoplasm. Both my sons, if you look at their cytoplasm, it's from their mother, not from me. That's why I resemble my mama. Now, whether you're light or dark complected, can that be controlled by daddy? Yeah, that can be controlled with pigmentation. But you gotta admit, all the cytoplasm you have right now can be traced back to one source, your mama. Or daddy didn't give you anything. When that sperm from your dad found the egg from your mom and they come together, and then the egg kept going, the egg cytoplasm, that became your cytoplasm. But half of you genetically is from your daddy. But physically, it ain't half and half. It's not really true. And you start thinking that way, you start getting some weird concepts going on. Alright? The number of chromosomes from one species to another one. Some ants only have 2. Only 2. Carp, that old ugly old fish, have 104. I got 52 pairs. They blow us away. And that carp, as you might know, we brought those here on purpose. That carp is not from America. We brought that sucker here. When did the Big Depression happen?
The Great Depression.
In the 20s. Folks are starving. So we said, well, there's a carp, there's a fish in Europe that them Europeans eat. It grows big, big, let's bring it here. And they did. And we don't eat the carp. And that carp, unfortunately, likes to bed where bass bed. Who is bigger, a bass or a carp? Carp is. Who's going to run who off? Carp's going to run bass off. Now, if you're a bass fisherman, is that good or bad for you? That's bad. We built a lake at my hunting land for my lost lake. And I put carp in that lake because I put grass carp in, but they've been sterilized. I don't want those suckers breeding in my lake. And I also bought and put in little baby bass. I bought and put in little baby catfish. Little bitty things. And I got a feeder going, and I fill the feeder up, and it looks like piranha eating under the feeder. And just last year, I caught an 8-pound bass in my lake. That was from those little bitty ones. Now, I ain't been fishing this year. Now, I intend to go until I can make a 10-pound bass, baby. I thought I'd hit a stump. And my line went sideways. I said, oh my God, that's no stump. There's a big old female bass. She's full of eggs, so I threw her back. There's a carp.
Wow.
And that's not camera angle. That's a big, ugly fish.
Are those the fish that jump out of the water when the boat?
No. They'll get by the bank and make the biggest noise you ever heard. They'll get in water too shallow. They can't get out. And you'll think there's a ton of fish fighting. That's a carp.
That's a mullet.
Who's that?
Tell you what, though, now, they're fun to catch.
So those are the 20 fish.
And a bow and arrow, I'll shoot them also. I got my arrow, and I got a line hook to it, I got a fish reel. I just put 808 on my bow, and before I had my leg gone, I was down on my boat. Of course, now where he is, you got to shoot above him because the water makes it bend, right? And that thing, boy, you start, it'll pull you off that boat. That's big now. I've never caught one that big. Now, the most anything has is 1,000, and that's by a plant. a fern has, this fern has 500 pairs. And see when I wrote down that word, Ophioglossum reticulatum, how I spell it? Because I can do it. You must put it in italics. Now, I didn't have to underline it. Now, you put in italics, all you got to do. But now, when I'm writing on a test answer, then I got to underline it. So if you're handwriting a species name, like we're Homo sapiens, when you write Homo sapiens, you better underline both words. Now, if you're typing it, you italicize both words. You don't do both. You don't. If you're typing it, now I did here just for teaching purposes, but in real life, that would not be underlined. It would just be slanted. But if I'm writing that, now also, see that second word there? The rule says, do not ever capitalize that second word. Homo, capital H, sapiens, small s, that's us. Homo erectus is our ancestor, the old caveman, the sloping forehead. They got a commercial on now about Neanderthal, that Neanderthal guy. It looks like caveman. That's Homo erectus. Looks like us, but don't have as big as brain case and not quite as smart. Now right here is how you really do it. If I'm typing it, I would do it this way. If I'm handwriting, I do it that way. And if you're handwriting it, you underline them. If you're typing it, you slant them. You don't underline it if you slant it. If you cannot slant it, then you must underline it. That's the rule for writing these scientific names. And it's not my rule. What time is it? I got to watch it.
You got a few minutes.
A few minutes.
There's only one more.
Let's see what. One more slide. All right. This is that fern with all those chromosomes and the dots. How where you find them? Anyone around Georgia, you reckon? No, you got. Where you got to go to find them?
Mexico.
South America, Panama Canal.
Africa.
But we're right here. And obviously, you're not going to walk outdoors to find a fern growing in your backyard. With this being the case, you think they like it warm and cold or hot and dry?
Hot and dry.
No. These are rainforests, buddy. Those are rainforests right there.
Australia.
Hot and warm. Wait, warm and wet. Warm, hot and humid. They like it hot and humid. But then all ferns like humid, right? But these bad boys. Now, the maidenhair fern, they're like a little fiddle. They like it cool and wet. You'll find those in Georgia. But that one that we mentioned right there, that Ophioglossum, don't go out looking, you won't find it. The number does not, I mean, you know that we're more complex than a stupid fern. So numbers don't mean nothing other they're just separate numbers. So don't even think about the more chromosomes, the more advanced. That is not true at all. And we'll come in next time I see you and start, I reckon that's 11.